Hello, everyone. Thank you very much for joining our webinar today. My name is Payo. I am a biotechnologist by training and have been working on the registration of products belonging to the food chain at Benentech Consulting for almost 9 years. I will be presenting today in relation to the requirements for the registration of microbial feed additives in the EU. Before we start, I wanted to let you know that you will receive a copy of all the slides and a copy of the recording of the webinar by email. We will also post a copy of the video recording on our YouTube channel. If you're having any problems with the audio or with the visuals, please use the chat function at the bottom of the screen, and one of my colleagues will be able to assist you. The webinar will be about approximately 45 minutes long, and I will address as many questions as possible at the end, and we'll be happy to answer any remaining ones by email. I wanted to thank everyone that has sent me questions in advance, and if you do have any additional questions during the presentation, please send these through, either using the chat or by email, and I will reply after the presentation. Right, so let's get started. During this webinar, I will first provide a brief introduction on microorganisms used in the food chain and on the regulatory framework. Then, specifically related to feed additives, we will outline the registration procedure and the different feed additive categories and functional groups belonging to each category. Finally, we will go through the data requirements for the characterization of microorganisms used as feed additives or as feed additive production strains. The whole genome sequencing plays a very important role in the strain identity and in the analysis of sequences of potential concern. We will also go through the requirements related to the antimicrobial resistance and antimicrobial production. Finally, for fermentation products, we will cover the possible carryover of either DNA or of the production strain to the final product. Microorganisms used in the food chain are generally bacteria, yeast, and filamentous fungi. However, there are other taxonomical groups used as well, such as phages or microalgae. Microorganisms can be genetically modified or not, and may be used either as such or as organisms of substances of interest, which could be vitamins, enzymes, or amino acids. The placement of such microorganisms or derived substances in the European market may be subject to a pre-market authorization process according to the relevant regulatory framework, which will be including a genetically modified food and feed, foods for which nutrition or health claims are made, food enzymes produced by fermentation, food additives, food flavorings, microorganisms used as plant protection products, novel foods, or feed additives, which is the framework that we will be focusing on today. So applicants that are interested in registering a feed additive should prepare a dossier in accordance with the requirements set in the applicable regulatory framework and also based on the EFSA guidance documents. In 2018, EFSA published guidance outlining the requirements for the characterization of microorganisms. A similar approach is also recommended for other products such as novel foods or food enzymes produced by fermentation. EFSA has also published a draft statement on the requirements for the whole genome sequence analysis of microorganisms intentionally used in the food chain. This statement was endorsed on the 4th of December of 2019 and was subject to a public consultation until the 28th of February 2020. The final version has not yet been released, but we recommend to follow the draft since EFSA is already taking into consideration the content for the applications that are currently under evaluation. During the webinar today, we will cover the key points from these two guidance documents. There are various steps involved in the registration of a feed additive. The workflow starts with a request for the EU authorization submitted by the applicant to the European Commission, who then forwards a mandate to the European Food Safety Authority. The applicant must send a copy of the complete technical dossier to EFSA together with some administrative documents. Declaration form and reference samples of the additive shall be sent to the European Union Reference Laboratory. They will be in charge of preparing an evaluation report on the analytical methods during the first 3 months of the evaluation in parallel to the risk assessment performed by EFSA. So EFSA is responsible for conducting a completeness check within 30 working days. After this period, a risk assessment is performed over 6 months. If necessary, EFSA will stop this clock and ask the applicant for further information during the assessment procedure. The clock will restart as soon as the supplementary information is received. Please bear in mind that there could be more than one clock stop during the evaluation procedure. So once the scientific assessment is completed, EFSA will adopt an opinion that is based on the information provided by the applicant. And based on this opinion, the European Commission decides whether to authorize or deny the authorization of the additive. The European Commission is assisted in this decision procedure by the member states within the animal nutrition section of the Standing Committee on Plants, Animals, Food and Feed. Finally, feed business operators are responsible for placing the feed additive on the market and for complying with any conditions or restrictions set by the legislation. Feed additives are used in animal nutrition for purposes of improving the quality of feed and the quality of food from animal origin, or to improve the animal's performance and health, for example, by providing enhanced digestibility of the feed materials. Feed additives are classified into 5 different categories. These categories are themselves divided into functional groups according to the additive's main functions. The authorizations granted for feed additives belonging to the category zootechnical additives and coccidiostats and histomonostats are holder specific, whereas the approval of substances belonging to the remaining categories, technological, sensory, nutritional, are not linked to a holder of this authorization. In any case, all of these authorizations are valid for 10 years and then are renewable after this period. The authorization process defines the need to prepare a technical dossier, which is divided into 5 sections. The main sections are those related to the feed additive's identity, which is in section 2, the safety for target animals, consumers, users and workers, and the environment, which is outlined in section 3. And efficacy of the microorganism for the intended use is included in section 4. These sections are built using the relevant guidance documents, which you can see in the slide. As I mentioned earlier, during this presentation, we will specifically focus on the guidance for the characterization of microorganisms used as feed additives or as production strains. Bear in mind that production strains can be genetically modified or not, but live microorganisms used as such are outside the scope of this guidance and would fall under the GMO regulation 1829 from 2003 rather than under the feed additive regulation, which is 1831 from 2003. The characterization of the microorganism is a critical part of the dossier, and will define whether the microorganism is safe and is suitable for registration or not. For this reason, we always advise to carry out these studies first before investing time and resources generating other studies such as the efficacy studies. A summary of the requirements for the characterization of feed additives containing viable microorganisms is shown on the left of this table. And a summary of the requirements for the feed additives produced by microorganisms are shown on the right. You can see also that the requirements vary depending on whether the microorganism of interest is a bacteria, a yeast, or fungi. And for production strains, the criteria also varies depending on whether the strains are genetically modified or not. As you can see, the identification is required for all strains. For bacteria and yeast, it should be based on the whole genome sequencing. Also, the assessment of the antimicrobial production, toxigenicity, and pathogenicity is required for all strains except for those that qualify as QPS. I will explain shortly what qualifying as QPS means for those that are not familiar with this concept. The antimicrobial susceptibility, based both on the genotype and on the phenotype, must be analyzed for all bacteria used as feed additives or as production strains. It is not required for fungi or yeasts. For production strains which have been genetically modified, a characterization of the modification is required. This should be done using the whole genome sequence for bacteria and for yeast, and of course for fungi if available. For all fermentation products, the absence of the production strain must be demonstrated. For fermentation products derived from either genetically modified microorganisms or from strains including sequences of concern, even though these may have not been genetically modified, the presence of DNA from the production strain must be evaluated. Finally, compatibility studies with coccidiostats or other antimicrobials may be required for feed additives containing viable strains. We'll go through each of these requirements in more detail. Just before going through each of these requirements, we will go back to the concept of QPS. So, qualified presumption of safety status provides a generic approach to the safety assessment of microorganisms, which are intentionally introduced into the food chain. It is not a concept that is exclusive to feed additives, but more applicable to any regulated microorganism. To justify that a microorganism is suitable for being evaluated according to the QPS approach, its taxonomic status should be established. It should be a species included in the QPS list, which is updated twice a year. And it should comply with any qualifications set in the QPS list. You can see here some examples of qualifications, which can be absence of toxigenic activity for Bacillus species, or in the case of Bacillus belacensis, absence of toxigenic potential and absence of aminoglycoside production. Those strains qualifying for the QPS approach are presumed safe for target species, consumers, and environment without the need for specific studies. This means that you would not need to conduct a target animal safety study or genotoxicity studies or environmental safety studies for these strains. The QPS concept applies also to microbial feed additives, which means additives derived from fermentation. So the QPS concept can be applied to genetically modified microorganism production strains if the parental or recipient strain qualifies as QPS, and also if the genetic modification is considered safe. In any case, as we mentioned earlier, the absence of DNA from the production strain must always be assessed for all products derived from genetically modified microorganisms. The whole genome sequencing provides the most complete genetic dataset for a reliable identification and characterization of microorganisms. These techniques are routinely used for bacteria and yeasts and are currently required by EFSA, even though these are not yet included in the regulatory framework. The whole genome sequencing is used for species and strain identification, for bacteria and yeast, and for fungi if available. The whole genome sequence is also used for the characterization of the genetic modification whenever relevant. And also it's used for searching for sequences of concern, including genes that code for or contributed to resistance to antimicrobials relevant to their use in humans or animals. And also for searches concerning genes related to virulence factors in the case of bacteria, and to known metabolic pathways involved in toxigenicity in relation to fungi. There can be challenges related to the use of the whole genome sequence due to, for example, the use of different sequencing technologies, which each have strengths and limitations. Also, the use of different approaches may give different results when analyzing the same data set, which could create difficulties in the interpretation. As experts are aware of the current limitations and will not look at the WGS analysis in isolation, but it will be looked at together with the phenotypic data in order to draw conclusions. In relation to the whole genome sequencing and to the reporting requirements, we're going to go through a few steps. So as a first step, the microorganism should be cultivated as a pure culture. Then the DNA is extracted, and the method for doing so should be described in the dossier. The sequencing step involves determining the order of nucleotides in a DNA molecule. The strategy and instrumentation must be documented, and according to the new draft WGS EFSA statement published in 2019, the trimming of the reads is recommended. The assembly step refers to aligning and merging DNA fragments in order to reconstruct the original sequence. This is needed when the DNA sequencing technology cannot read the whole genomes in one go. So if an assembly-based approach is taken, then we must provide a description of the approach, the software, the version, and the parameters used. A statistical measure of the sequence quality must be provided. A contig is a set of overlapping DNA segments that together represent a consensus region of DNA. The total length of the contigs relative to the expected genome size needs to be calculated and reported in the dossier. In relation to the DNA annotation, this is a process of identifying the locations of the genes and of all of the coding regions in a genome and determining their function. If genome annotation is carried out, then the software name, the version, and the parameters used should be reported together with the number of genes and pseudogenes identified. For fungi, information on the quality of the annotations obtained from relevant databases must be reported. And finally, the FASTA files of the whole genome sequence is required according to the current 2018 guidance. However, according to the new draft statement on WGS, it appears that FASTQ files will be required as well. These files are considered like raw data of the genome. All microorganisms under assessment should be identified unambiguously at species level based on up-to-date methodologies and current knowledge. The whole genome sequence analysis is required for the characterization of both bacteria and yeast, as you can see in the table. Also for any types of microorganisms, either used as feed additives or as production strains. For bacteria, it is preferable to use techniques such as average nucleotide identity, phylogenomic methods, or digital DNA-DNA hybridization. Alternatively, sequences such as the 16S RNA gene or other housekeeping genes can also be compared to relevant databases. From our recent experience with EFSA, we have observed that sometimes the analysis of the 16S sequences alone is not considered sufficient. So therefore, we recommend using phylogenomic methods. For yeasts, the taxonomy can be established by phylogenomics, by comparing specific sequences to relevant databases, or by alignment to a complete genome from the same species. Finally, for filamentous fungi, whenever the whole genome sequence is available, the identification should be made by phylogenomic analysis. If not, it should be done by comparing the 18S RNA gene and or the ITS regions or other characteristic genes with sequences deposited in databases. The strain needs to be deposited in an internationally recognized culture collection center, preferably in the European Union, and the certificate should be valid for the whole period of the authorization of the additive. If a production strain is genetically modified, then the genetic modification should be described. The applicant must indicate what is the purpose of the genetic modification and the characteristics of the modified sequences. For bacteria and yeast, the structure of the genetic modification must be addressed comparing the WGS from the genetically modified strain with the one from the non-modified parental or recipient strain to identify the differences between the two. If the WGS data is not available, for example, for fungi, an applicant should describe the steps followed in order to identify all of the genetic material that has been altered. For bacterial strains belonging to a species not included in the QPS list, the whole genome sequence analysis should be used to identify genes coding for known virulence factors, for example, toxins, invasion factors, or adhesion factors. For this purpose, comparison against up-to-date databases should be performed. There are strains for which the safety can be established by specific tests, for example, Enterococcus faecium and Bacillus species for which a cytotoxicity study is required. For eukaryotic microorganisms, the potential pathogenicity or ability to produce metabolites that could be harmful to humans or animals should be assessed. Targeted searches can be done to identify the presence of known metabolic pathways involved in toxigenicity. even though the current 2018 microbial guidance does not establish any reporting threshold values, the new EFSA 2019 draft indicates that hits in the database with at least 80% similarity at protein level or equivalent and 60% coverage of the query sequence should be reported. If there is an expected hit against a gene in the database, then further investigation may be needed. In this presentation, I have included the information from the new 2019 WGS statement in red, since these recommendations are provisional and may change, but we still recommend to follow them for now as a reference. The whole genome sequence of bacteria should be interrogated for the presence of genes coding for or contributing to resistance to antimicrobials. For this purpose, comparison against up-to-date databases should be performed. The new EFSA draft WGS statement from 2019 indicates that the searches must be done against at least 2 databases. So even though the use of 2 databases is not required according to the 2018 current guidance for feed additives, we do recommend following this approach. It may also be helpful towards the interpretation of the results. The current EFSA 2018 microbial guidance again indicates no thresholds for the genome AMR analysis. However, the new WGS draft statement from 2019 indicates that hits in the database with at least 70% similarity at protein level or equivalent and 60% coverage of the query sequence should be reported. It is very important to bear in mind that the genome data related to antimicrobial resistance is not interpreted in isolation. The results from these bioinformatic analysis are looked at together with the outcome of the phenotypic testing, as we will see over the next couple of slides. As you know, phenotypic antimicrobial resistance needs to be assessed for all bacteria, either included as viable cells in feed additives or used as production organisms. Microbial feed additives should not add to the pool of antimicrobial resistance genes that are already present in the gut bacterial population or increase the spread of antimicrobial resistance. Intrinsic resistance is inherent to a bacterial species and is typical of all the strains of that species. Intrinsic antimicrobial resistance is not considered a safety concern. Resistance to a given antimicrobial in a strain of a typical susceptible species is regarded as acquired, which does require further investigation and could be considered a safety concern. In order to decide whether a specific resistance is intrinsic or acquired or a safety concern or not, 2 different sets of data are analyzed. First, the genome data for the presence of known antimicrobial resistance genes as described in the previous slide. And secondly, the phenotypic testing based on the determination of a minimum inhibitory concentration for a selected group of antimicrobials. So when we look at the 2 sets of data together, we can see several scenarios. If the phenotypic resistance can be directly related to the presence of a known antimicrobial resistance gene, this is considered a hazard, unless the resistance is regarded as intrinsic, which means that it is present in other strains belonging to the same species. In order to make this assessment, it also may be necessary to perform searches in the literature. Secondly, if phenotypic resistance cannot be linked to the presence of a known antimicrobial resistance gene, there results will be reviewed on a case-by-case basis, but generally no further studies would be required. If the genetic analysis reveals antimicrobial resistance genes for antimicrobials of concern, but these are not expressed phenotypically, which means that the MIC values are below the reference values set by EFSA, the likelihood of the genes becoming active should be assessed either through in silico or in vitro studies. Finally, strains that are phenotypically susceptible to antibiotics and show no antimicrobial resistance genes of concern in the genetic analysis would not be considered a hazard by EFSA and would be regarded as safe. In relation to antimicrobial production, unless a strain qualifies for the QPS approach, or belongs to a taxonomic unit known not to produce relevant antimicrobials, testing should be made to assess the inhibitory activity of culture supernatants against reference strains that are known to be susceptible to a range of antibiotics. If there's a positive outcome in one or more studies, in one or more species, the inhibitory substance should be identified. And for the production strains for which antimicrobial activity has been identified, the absence of carryover into the final product needs to be evaluated. In relation to the presence of recombinant DNA, it must be determined for products obtained using genetically modified production strains, or for products obtained using non-genetically modified production strains that carry acquired antimicrobial resistance genes or other potential genes of concern. This analysis must be done by PCR techniques, and they should be done in 3 independent batches analyzed in triplicates. The limit of detection should be 10 nanograms of DNA per gram or milliliter of product or lower. It is very important to follow all of the requirements and indications included in the EFSA guidance for these type of tests, including the different control groups or the lysis step, etc. The absence of the production strain in the final product must be analyzed for all feed additives produced by fermentation. The test can also be done in a more concentrated unformulated precursor, and then the results can be extrapolated provided that the test item is representative. The absence of viable cells should be investigated in 3 lots in triplicate again through culture-based method. It should enable the recovery of stressed cells by cultivation in media with a minimal selective pressure. It can also be assessed by providing a longer incubation time compared to the normal culture time, for example, at least double of the time. So finally, we're going to bring all the data together generated either in silico or in vivo, including taxonomy, antimicrobial resistance, antimicrobial production, toxogenicity, absence of DNA or production strain in the final product, in order to draw the conclusions on the product safety. So first, starting with feed additives that contain viable organisms. For strains qualifying for the QPS approach to safety assessment, no risks are identified for target species, consumers, and the environment. Here is an example of a positive opinion that EFSA issued for a microbial QPS additive. You can see that this strain was not considered toxigenic, which is one of the QPS qualifiers for Bacillus species, and also no antimicrobial resistance of concern was found. So the opinion was positive. On the other hand, bacterial strains carrying acquired antimicrobial resistance genes are considered to represent a risk. Also, other pathogenic, virulent, or toxigenic strains, and those capable of producing relevant antimicrobials, would be considered a hazard and are very unlikely to get authorized. Here you can see an opposite example of a negative opinion issued by EFSA for a microbial additive with acquired antimicrobial resistance genes identified. This product has been unfortunately removed from the market. Finally, for bacterial strains that, in spite of not qualifying for QPS, are free from acquired antibiotic resistance determinants, as well as for other strains that are not known to produce relevant antimicrobial substances and are not pathogenic or toxigenic. These strains are considered safe for the target species, the consumers, and the environment. In all cases, user safety should be addressed separately according to the relevant guidance. And of course, other parts of the dossier need to be constructed according to the relevant guidance, such as efficacy or the identity guidance. But again, it is really critical to establish the safety of the microorganisms as a first step. Now moving on to strains used for the production of feed additives. For the strains that either qualify for the QPS approach or are free from antibiotic resistance determinants, do not produce relevant antimicrobial substances, or are shown not to be pathogenic or toxigenic, and for which there is no genetic modification of concern, no risks are expected to arise from the metabolism of the production strain itself. Other microbial strains that produce feed additives could be toxigenic and or able to produce antimicrobials, and these would be considered a risk, unless the absence of the relevant toxins and or the antimicrobials can be demonstrated in the final additive. Finally, bacteria carrying acquired antimicrobial resistance genes used as production strains would also be considered to represent a risk for target species and to those exposed to the additive, unless the absence of the DNA from the production strain can be shown in the final product. In all cases, further aspects of the safety of the product for the target species, for the consumers, for the users, and the environment should be considered separately according to the relevant guidance. Here you can see on the right an example of a positive opinion on a product derived from fermentation in which the DNA and the production strain was not detected. On the contrary, on the left, you can see an example of a product for which the production strain and the sequences of concern were detected in the final product, and therefore this was considered a risk and the EFSA opinion was negative. To wrap up, in relation to the strains used for fermentation, if these include sequences of concern or produce substances of concern, it is critical that there is no carryover to the final product. And just to reinforce again, in relation to fermentation products, that the strain characterization is a critical first step for the registration of the feed additive, and these types of studies should be carried out first before performing the remaining studies required in the EU dossier. We're going to end the presentation with a brief questions and answer session. Thanks again to all of you who submitted these questions while registering to the webinar. I have selected a few in order to cover a range of different topics, and I will reply to any remaining ones by email. The first question is related to the acceptance of the GRAS status in the EU and to whether strains can be registered individually or not. as well whether the registration studies must be performed with individual strains or can be done in combination. So in relation to the first question, no, the generally recognized safe status does not apply in the European Union, and it is not recognized by the European Food Safety Authority or any of the EU authorities. In the US, our understanding is that direct fed microbials must be registered individually. However, in the EU, strains can be registered either individually or in combination. Strains must always be individually characterized, but efficacy can be tested per strain if these will be registered individually, or tests can be done with a preparation if multiple strains will be registered together. If a combination of strains are registered throughout one application, these won't be allowed for individual use. The registration of individual strains may be an advantage if there's an interest in mixing and matching strains in different products. The next question is related to keeping the WGS data confidential and about the changes expected due to the new transparency policy. So currently the European Commission decides on the confidentiality, whereas after March 2021, once the new regulation on transparency enters into force, it will be EFSA taking this decision. In both cases, the EU authorities will grant confidential treatment to proprietary information and will certainly agree to keep the WGS data as confidential. If you would like to know more about the transparency regulation, I invite you to watch the webinar that my colleague Eliana Enrique delivered on this topic. You can watch it on our YouTube channel, or you can also email me and I will send you a copy of the recording. The next question is related to whether efficacy studies can be performed outside the European Union and whether these must be conducted at a GCP accredited center or not. Microorganisms can be used in animal nutrition to achieve an effect on the feed itself, for example, as acidifiers, as preservatives, or hygiene condition enhancers. They can also be used to exert an effect on the animal, such as an improvement of performance, for example, as a result of a beneficial effect in the gut flora. For additives that affect the characteristics of the feed, the efficacy should be demonstrated using laboratory-based studies. In vivo animal studies are foreseen for all additives for which the intended effect is seen in the target species. So in the case that in vivo studies are needed, a minimum of 3 independent in vivo studies showing significant effects should be provided for each species. These should be carried out at least at 2 different locations, and one of them should be in the European Union. But the remaining 2 studies could be done outside the EU, but following representative feed practice. All trials should follow the criteria established by recognized externally audited quality assurance schemes, for example, good laboratory practice or good clinical practice, but this does not mean that the centers need to be accredited. It just means that they should follow specific quality assurance criteria. Right, so the next question is related to user worker safety and to whether the inhalation toxicity can be avoided for microorganisms with, which have a small particle size. So a microorganism that qualifies as QPS is considered safe for target species, consumers, and environment, as we said during the webinar. However, user worker safety must be considered separately. Due to the product nature, Microorganisms are presumed respiratory sensitizers, and standard precautions when handling the product are advised. For example, the use of masks or other protective equipment. Generally, microorganisms do not have a potential for serious adverse effects at low levels of exposure that would require inhalation toxicity testing. However, skin and eye irritancy and sensitization would need to be evaluated. I'm sharing here in the slides a couple of examples of EFSA opinions, which you can see. In which EFSA has drawn conclusions on inhalation toxicity for microbial products, either with high or with low dosing potential. For those of you that are very familiar with the EU feed additive guidance documents, you will know that all of the guidance documents related to feed additives were updated during 2017 and 2018, except for the guidance on user worker safety. This is because there is a very active, lively debate between the member states, the European Commission and EFSA in relation to the requirements. Therefore, we should expect changes to come on user worker safety. In the past, in the absence of user worker safety data, EFSA drew assumptions based on the worst case scenario and recommended standard precautions. However, the lack of data scenario does not seem to be accepted anymore. The next question is related to the WGS statement and their interpretation of the results from the CARD database. Normally, after a public consultation, EFSA reviews all the comments and questions, addresses them, and then adopts the final version. This process normally takes around 2 to 3 months. This specific public consultation on the whole genome sequencing ended on the 28th of February, and the final version should be published soon, although I'm afraid we still don't know the exact date. I do assume it's taking longer than usual, as the scope of the statement is very wide, and besides feed additives, it affects many regulatory frameworks such as food enzymes, food additives, food flavors, plant protection products, novel foods, feed additives, nutritional health claims. Also, the number of inputs that they receive must have been very high. In relation to the interpretation from the bioinformatic searches with CARD analysis, intrinsic genes are those present in all strains within a species. To interpret the results, the outcome of the genomic analysis must be combined with the phenotypic analysis and put into context. For example, if a bacillus species strains shows resistance to streptomycin and is associated to genes of potential concern, then we will need to look at whether these traits are present in other strains in the same species and genus, also whether the strains are complete or are partial, and whether there is any risk of horizontal transfer potential. Another example is related to the strain including a gene related to antimicrobial resistance. It may be considered acquired if it's not present in other strains belonging to the same species. The analysis of mobile genetic elements is not a specific requirement by EFSA because they presume that all of the elements may be potentially transferable. The last question is related to advice on how to avoid EFSA clock stops, and our main advice is to always follow the EFSA guidance. If you follow very closely the most recent EFSA guidance, you will have less risk of receiving questions from EFSA, and ultimately you will achieve a quicker time to market, and you will also save costs. Some of the common questions received recently from EFSA related to microorganisms are related to the strain deposit certificate. So if a strain was deposited many years ago, you may be asked for a new certificate or for proof that the current one is still valid. In relation to taxonomic identification, as we have discussed in the webinar, certain techniques are required based on the whole genome sequence for bacteria or for yeasts. So if different techniques are used, EFSA will likely ask for a repetition of the taxonomic analysis. In relation to antimicrobial protection or susceptibility or toxigenicity when relevant, EFSA will insist that these studies are performed according to the indications in the current guidance. As for the sequences of concern derived from the analysis of the whole genome sequence, they should also be adequately reported. We advise to use the thresholds discussed during this webinar, which are 70% identity for antimicrobial resistance genes and 80% of identity or higher for virulence factors. If you, for example, submit a report showing only perfect hits, it is likely that EFSA would question the outcome of the reports. In relation to the absence of DNA in the final product, it's important to follow all of the steps indicated in the EFSA guidance. EFSA could question or ask for a repetition of results if the LOD is not adequate, if the number of batches or replications is lower than required from the guidance, or if adequate controls were not used. Also, for the analysis of the production strain in the final product, it is also important to follow the guidance in relation to the use of selected media or culture time, the number of samples. Otherwise, EFSA may also question the data. This was the last question selected, and we're going to wrap up for today. I will make sure to contact you to reply to all of the remaining questions received. Thank you very much for joining. I hope you found this webinar useful. If you are interested in watching other webinars related to regulatory aspects of the food and feed chain, please feel free to visit our website or our YouTube channel. Also, if you need any regulatory assistance, please feel free to contact us. And thanks again for joining, and hope you all have a lovely weekend.